Hello everyone. Today in this video lecture we are going to study about embryology of nervous system. Now in this video lecture I will tell in detail how nervous system is formed. Firstly, let me define what is nervous system. Nervous system is a system which make a human, or we can say which make an organism to have some functional relationship with its environment. I am stressing on the word functional relationship. So system which connects an organism to the environment via the functional relationships. So this include nervous system. So when an embryo reaches 20th day of intrauterine life, so there you can see 3 germinal layers, 1, 2 and 3. The uppermost is known as ectoderm, the middle one is known as mesoderm, and the innermost one is known as endoderm. Now these 3 layers are formed in the fetus life at 20th day of intrauterine life. So as generally animals are triploblastic, we mammals, so this ectoderm has a groove here, and this groove is known as neural groove. Which groove? Neural groove. Now what happens after these 20th day? What happens? This neural groove starts forming a tube. It starts forming a tube like thing, and in the center of this we can say neural pore, and above this neural pore there are certain cells. These cells are not the part of neural tube. These are known as neural crest cells. And this is the neural tube. So in the embryonic life we see these neural crest cell and neural tube in the ectoderm. Now this neural crest cells, they give rise to peripheral nervous system, and this neural tube give rise to central nervous system. Now this neural crest cell give rise to peripheral nervous system. Now what is this peripheral nervous system? Peripheral nervous system comprises of all the nerves associated with the central nervous system. Now central nervous system is formed by the neural tube, this neural tube. Now this neural tube forms the central nervous system. And what is this central nervous system? It is the system comprising of brain and spinal cord. Now what happens? You are seeing this neural pore. In this neural pore, in the embryonic life, there is a fluid known as amniotic fluid. Which fluid? Amniotic fluid. And in the baby, when a child is born, this amniotic fluid gets changed into cerebrospinal fluid, which flows through the ventricles of the brain. Okay, so it is the precursor to CSF, cerebrospinal fluid, which flows into the neural pore in the embryonic stage. Now what happens? As PNS is clear to you that PNS, peripheral nervous system, is formed by the neural crest cells, which are the cells between the ectoderm and neural tube. Now what happened to this neural tube? This neural tube forms certain vesicles. It forms certain vesicles. Now if you will see, there are certain primary vesicles. Primary vesicles includes prosencephalon, mesencephalon, and rhombencephalon. Now this prosencephalon in the embryonic stage changes to forebrain in the baby. This is nothing, prosencephalon is nothing but the forebrain, and mesencephalon similarly is the midbrain, and rhombencephalon is hindbrain. So neural tube changes into three vesicles, which includes prosencephalon, mesencephalon, and rhombencephalon. This prosencephalon is forebrain, this mesencephalon is midbrain, and this rhombencephalon is hindbrain. Now what happens after some time? This prosencephalon further divides into diencephalon, like these two things. These are diencephalon, and this is our telencephalon. Now this diencephalon includes thalamus, hypothalamus, and epithalamus, and telencephalon includes cerebrum. Similarly, this midbrain is similarly midbrain, and this rhombencephalon gets changed into metencephalon and myelencephalon. This metencephalon is what? This is the secondary vesicles, and this diencephalon, telencephalon, these all the things which I have written in this line are the secondary vesicles, and things written on this side are the primary vesicles. So this rhombencephalon or hindbrain gets changed into metencephalon, which give rise to pons varoli and cerebellum, and this myelencephalon gets changed into medulla oblongata, which forms posteriorly spinal cord. So in an animal, what we see, let me explain again it for you in a jiffy. We have three germinal layers: ectoderm, mesoderm, and endoderm. This ectoderm has a neural groove. This neural groove at a particular stage forms the neural tube, and above this neural tube you will find neural crest cells. These neural crest cells form the peripheral nervous system, and this neural tube forms the central nervous system, which includes brain and spinal cord. In this neural tube you will find the neural pore, where cerebrospinal fluid is present in its precursor form known as amniotic fluid. Then this neural tube after some times gets changed into some primary vesicles, three primary vesicles: prosencephalon, mesencephalon, and rhombencephalon. After some time this prosencephalon gets changed, these primary vesicles gets changed into secondary vesicles. So prosencephalon gets changed into diencephalon, or we can say divided into diencephalon and telencephalon. Diencephalon is nothing but a accumulation of thalamus, hypothalamus, and epithalamus. Epithalamus is generally not visible, and this telencephalon is nothing but cerebrum, including cerebral cortex joined by corpus callosum. Now after that we have midbrain. Midbrain is formed by mesencephalon, and then we have rhombencephalon, which gets divided into metencephalon, forming pons varoli and cerebellum. Then this myelencephalon forms the medulla oblongata, which forms posteriorly what thing? Spinal cord. Now in the neural tube there are certain cells which are known as neuroepithelial cells. Which cells? Neuroepithelial cells. Neuroepithelial cells in the neural tube are nothing but stem cells. These neural tube cells or neuroepithelial cells forms the neuroglia, which are the sporting cells, and neurons also. This was an additional point which I told you. So this was all about the embryology of nervous system. In the upcoming video lectures, we will study in detail about leptomeninges and other kind of things like cerebrum, forebrain, midbrain, and hindbrain, as well as spinal cord. Thanks.